Next, we have biochemical test. This is Luciferase assay. Luciferase is an enzyme that produces light, and this light is directly proportional to the ATP production by the bacteria. This test is used for the detection of drug-resistant MTB. So, if the bacteria is resistant to the drug, it will not get killed by the drug, and it can produce the normal amount of ATP. So, the luciferase assay will produce normal amount of light, but if the MTB is sensitive to the drug, it can get killed by the drug and it cannot produce a normal amount of ATP. So less amount of ATP is produced and the luciferase assay will give less amount of light. The next is immunological test. This is the procedure that occur in both these tests, that the macrophage engulfs the bacteria. The macrophage present the antigen on the MHC class to become the antigen presenting cell, and the macrophage will activate the T cell and the T cell will release the IFN gamma. Remember that this IFN gamma production is calculated in the interferon gamma release assay IGRA. It has two versions, Quantiferon-TB gold and the T-spot TB. The basic difference between them is that in Quantiferon-TB gold we measure the concentration of IFN gamma in the whole blood, while in T-spot TB we count the individual T cell that are producing this IFN gamma. In tuberculin skin test, which is also known as PPD or Mantoux test, it is a type of delayed hypersensitivity reaction type 4. The basic procedure is we give intradermal injection of purified protein derivative. The purified protein derivative is TB antigen, and if the person is previously infected with TB, the memory T cells that remember that TB can recognize the PPD2 and will release the cytokines, the IFN gamma. At that place, the recruitment of macrophage and other immune cells will take place, and in duration, this reaction will occur, and in duration, and swelling will form at the place, and that can be measured after 48 to 72 hours. Also, remember this point that in case of latent disease and active disease, you can get both IGRA and TST test positive in latent and active disease. In latent disease, you cannot see the visible symptoms, while in active disease, you can see the visible symptoms. But in case of BCG vaccination, the vaccination for TB, tuberculosis, you can get a false positive tuberculin skin test positive in case of BCG vaccination, while in the IGRA will remain negative in case of BCG vaccination. So BCG vaccination can give you a false TST test positive.